हेलो हेलो।
हम्म,
हाँ सर,
हेलो।
लाइव हो गया, लाइव चालू हो गया।
लाइव चालू हो गया ना?
यस यस।
शुरू करें।
हाँ शुरू कीजिए, शुरू कीजिए।
हेलो एवरीवन, गुड इवनिंग।
वेट सर।
ओके।
सर कैन बी स्टार्ट सर?
या
एक मिनट सर, एक मिनट। Sir, I'm going to start a session, sir. Sir, good
Okay.
evening. Evening, all the delegates. Today we are gathered for webinar in discussion on rabies and anti-rabies vaccine. Sir, thank each and every all of you for your still present at the occasion. Now, I would like to welcome our respected speaker, Dr. Vijay Kumar, who is a renowned physician and always care about the prevention from the rabies. Now, I am going to hand over the session of Dr. Vijay Kumar. Sir, please start the session, sir.
hello everyone, good evening. Today we are talking about a new anti-rabies vaccine.
Thank you, sir.
But first we have to know about the rabies. As we all know, it is a zoonotic viral disease that is caused by rabies virus. And it claims highest fatality among all infectious diseases. But it can be prevented by vaccination. In past, we have seen many effective vaccines were developed that used either live attenuated strains of a pathogen or inactivated killed pathogens. Live attenuated vaccine strains are typically highly immunogenic but has inherent safety concerns. There are so many side effects or even death. The inactivated or killed vaccine which we use stimulate a weaker immune reaction, and thus they may require the administration of multiple doses. An important practical limitation and increases non-compliance rate. As described by Sankaraya, the non-compliance rate increases with increase in each dose, and it was 40% at the 5th dose. That is very high. The development of rabies vaccine considered through 4 chronological stages, which are neural tissue vaccine, non-neural tissue vaccines, cell culture-based vaccines, and now recombinant DNA and protein-based vaccines. During these development processes, various killed, inactivated, and live attenuated vaccines were tested. From these studies, it was observed that killed virus vaccines were less efficient due to poor immunogenicity and lack of a strong T and B cell response. The members of the first generation rabies vaccine that were vaccines prepared in neural tissues included Pasteur vaccine that was in 1885, live attenuated, Hoes vaccine 1887, live attenuated again, and Pasteur vaccine that was 1895, inactivated vaccine. Then came suckling mouse brain vaccine 1955, suckling rat brain vaccine 1955 again, and suckling rabbit brain vaccine that is in 1955. That was also inactivated vaccine. All these vaccines promote autoimmune reactions in the recipient, some serious side effects were also observed, like neurological problems and even death. So, due to these reasons, the second generation non-neural vaccines were developed, where we used embryonated eggs for culturing the virus. Later, the virus was inactivated and used as vaccine. Sorry. These vaccines were administered by intramuscular route in the deltoid muscle or in the thigh muscle in case of small children. Total of 6 injections over a period of 90 days were given for protection. Although these vaccines were safer than the previous generation vaccine, or we can say first generation vaccine, the major disadvantage of these vaccines was the need for large number of doses to induce sufficient protection beside their serious side effects. Now we come to the third generation. The third generation of vaccines were developed using animal cell culture technique, where virus was cultivated in hamster kidney cells and human diploid cells. The third generation vaccines were economical compared to the first and second generation vaccines. However, these vaccines are expensive due to requirement of multiple doses. That increases their overall cost. The major disadvantages of this vaccine is poor immune responses in malnourished animals or humans, even humans. Now we come to the fourth generation of rabies vaccine. These are developed based on recombinant DNA technology, where either the DNA vaccine or the protein subunit vaccine is used. Now they are being developed. Rabies virus contains usually 5 genes, that is nucleoprotein, N protein, phosphoprotein, that is P, matrix protein, that is M, glycoprotein G, and the viral RNA polymerase, that is L protein. Among these proteins, the G and N proteins are known to be important for immunogenicity against rabies virus. So here we choose G and N protein that are very important. Regarding the DNA vaccine, unfortunately in humans, the DNA vaccines were found to be poorly immunogenic. So we can't use them. Now we come for the rabies G spike proteins. They play an important role in the virion binding to host cells through a specific recognition of the cell surface receptor molecules. It is the major antigen in the formation of neutralizing antibodies against rabies. Studies indicate that the rabies G protein plays a crucial role in pathogenesis of rabies and is the most antigenic and immunogenic surface proteins involved in viral attachment. Virus neutralizing antibodies exclusively binds to the rabies G protein. Sorry. Here, this point is very important. Antibodies bind to the G protein, exclusively bind to the rabies G protein. The G protein activates the CD4 T cells, which are vital to release long-lived antibodies in the blood. Therefore, G protein can be considered the ideal human anti-rabies vaccine. Therefore, for the development of subunit rabies vaccine, strategically, SF9 cell line is used to produce native proteins as particles, commonly known as virus-like particles, that is VLP. This is the Thrabies. Is the first novel recombinant rabies G vaccine prepared by using VLP technology. Now you can see what is this VLP technology. A very powerful method for developing vaccines. VLPs are multi-subunit self-assembly competent protein structures with identical or highly related overall structure to their corresponding native viruses, which doesn't carry any viral genetic material such as nucleic acid or infectious viruses. The non-infectious nature of VLP. The non-infectious nature of VLP, because it doesn't contain any viral genetic material like nucleic acid or infectious viruses, significantly improves their safety profile over live attenuated vaccine. Another form of subunit, killed or particulate vaccination. VLPs have great potential for a number of applications in addition to vaccines. Now we go for VLP structure. VLPs are supramolecular assemblies with the same or similar structure as native variants of about 10 to 200 nanometer in diameter. VLPs are made up to copies of one or more viral proteins that self-assemble into nanoparticles. VLPs doesn't contain, again I repeat it, VLPs do not contain any genetic material, therefore non-infectious to vaccinated individuals and safer than whole pathogen-based vaccine, such as those containing live attenuated vaccines or viruses. Due to the above, VLPs do not have the drawback of replicating, recombining, reassortment, or reverting to virulent stage as may occur with traditional vaccines. That is very important point, and that is very strong point. In terms of immunity, VLPs represent pathogen-associated molecular pattern, PAMs, due to their multi-matrix substructures with conformation that resemble that from wild-type viruses. VLPs can be discriminated against by the host and therefore may induce an adaptive immune response. Now we come immune responses induced by VLPs. VLPs can directly interact with antigen-presenting cells. They are sensed by pathogen recognition receptors, which are expressed on the surface or endosomes from dendritic cells. They present them as antigens. VLP uptake prompts DC maturation, dendritic cell maturation, and presentation of peptides loaded into major histocompatibility complex class 1 or class 2 molecules for priming CD8 or CD4 T cell responses respectively. CD4 T cells help B cells to produce antibodies and enhance CD8 T cell cytotoxicity. So here both humoral and cellular immunity gets activated through antibodies and CD8 plus T cells. Since VLPs contain multimeric epitopes on their surface, they can promote cross-linking of B cell receptors. Simple cross-linking of BCRs, B cell receptors by VLPs can be strong enough for priming B cells and induce the production of antibodies through plasma cells, even without the help of CD4 T cells. So it can act directly to the B cells without mediating CD4 cells. B cells can work as APCs, antigen-presenting cells, taking up VLPs, processing and presenting them to the T cells. Therefore, VLP-based vaccines are capable of inducing both humoral and cellular immune response. Due to their multimeric nature, adjuvant co-administration is not needed in most cases. But the use of adjuvant improves their immunogenicity. This is also true. Now, what are the disadvantages of VLPs? The traditional attenuated vaccines contains pathogens that can replicate into the host cells, may lead to various side effects, sometimes may result in new pathogenic strains. That is important. That is the mutant variety. Like traditional vaccines, VLPs are immunogenic and excellent to control infectious disease. One of the main advantages of VLPs is safety. VLPs cannot replicate, recombine, or undergo reassortment because they do not contain any infectious DNA or RNA material. Therefore, VLPs are safer than traditional vaccines. VLP-based commercially available vaccines are produced in yeast and baculovirus expression systems. Baculovirus are often found in vegetables, and fortunately, they are not capable to replicate in mammalian cells. Therefore, they are not pathogenic for humans or mammals. VLPs possess several advantages over products that are produced by chemical synthesis, such as a smaller size, which ranges from 10 to 2000 nanometer, availability of high resolution, three-dimensional models of their structure, construction flexibility, high production yields, and a structural uniformity of each type of virus or VLP. The VLPs are stable, unlike the virus, at room temperature for over 6 months and the immunogenicity is as potent as viral vaccine. That is important. Now we come to the limitation. But currently VLPs are as good as traditional vaccines, but with the advantage of being safer. However, some drawback needed to be highlighted. One is, since vaccine based on VLPs are very complex and therefore their price is a bit high. VLPs require a purification process where the use of density gradients or even chromatography are necessary, and therefore the cost of this downstream process is high and a bit time consuming. Now we come to the vaccine part. Recombinant rabies G protein vaccine provides a collective strength of multiple binding sites, avidity, and thus improved antigenic stability and immunogenicity. Since it is produced being recombinant technologies provides better control over antigen content and immunogenicity compared to inactivated virus containing vaccines available currently. So we can control the content of antigen, and so the safety profile becomes very high. Currently, 5 dose regimes administered on day 0, 3, 7, 14, and 28 are recommended for post-exposure vaccination, post-exposure prophylaxis of rabies. The NOVE is the first 3-dose rabies vaccine, that is 037, in the deltoid muscle, which may help to improve compliance towards full course of immunization due to unique advantages like reduced doses schedules, less number of injections, and reduced number of visits to clinic or hospitals. So here we come, safety of rabies vaccines are prime concern because of the near 99% mortality rate of infections with pathogenic virus systems. For this reason, only killed vaccines have been approved for use in humans. Thus, numerous approaches have been pursued to develop alternatives to traditional live rabies vaccines. Recombinant rabies vaccine protein rabies is developed by using VLP technology. VLP are considered as safe and effective candidate of therapeutic vaccines. Therefore, this rabies can be considered the ideal human anti-rabies vaccine with improved antigen stability and immunogenicity. Thank you. So any questions? Any questions regarding rabies vaccine or rabies?
Any question regarding
rabies? Rabies or animal bites or something like that? Okay, we can conclude then.
Any questions?
Huh?
सर एक क्वेश्चन ये है
बोलिए
सर, एक क्वेश्चन है कि जो पेशेंट दूसरे कंपनी के एंटी रेबीज लिया हुआ है सर, 2 डोज लिया सर, और तीसरा डोज अगर लेना चाहे हम लोग का तो उसमें शिफ्ट कर सकता है कि नहीं सर पैटर्न?
Regarding immunogenicity, it will be compatible, but he can take Yeah, you can take.
ले सकता है सर.
Yeah, ले सकते हैं. एक क्वेश्चन मैं देख रहा हूं इधर. Is it about
Question तो नहीं दिख रहा है
pre-exposure
सर अभी.
prophylaxis? हाँ, ये मेरे ये प्यारा है. For post pre-exposure prophylaxis, that is for dog handlers, veterinary doctors, or employees. There is no current data about any pre-exposure prophylaxis of rabies. So we can't recommend for pre-exposure. This is only right now. This is only for post-exposure prophylaxis. For pre-exposure, we still go for HDC vaccines.
Any question? सर ओवर करते हैं सर मीटिंग को सर. Sir, first of all, sir, many thanks for Dr. Vijay Kumar. Their valuable speech, which was very presentable. Message is very clear that 3-dose anti-rabies is more effective as compared to 5-dose vaccine available in the market. On behalf of Cadila Pharmaceuticals and Rabies, I thank you all for taking time out of from your busy schedule. Thank you, sir. Thank you so much for your
Thank
valuable
you.
support,
Thank you,
sir.
Chalo. Bye.
Thank you, sir.